Dear students, today we will be studying about another cell organelle, that is the Golgi apparatus. It is also known as Golgi bodies. In the earlier lecture we have studied about endoplasmic reticulum. It was of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Now we will continue with the Golgi bodies. So Golgi body was discovered by Camillo Golgi and has the name Golgi bodies or Golgi apparatus. Again, just like the endoplasmic reticulum, Golgi apparatus or the Golgi bodies are also membrane bound structures. Single membrane is present and they occur in three forms, that is the tubules, vesicles or cisternae. We will just see it in the next slide in the form of the diagram. And as I told you earlier that ER was also surrounding the nucleus, this Golgi apparatus is also seen aggregated towards the nucleus only. In case of the plants, they are unorganized and they are not connected with the help of the channel-like structures, and they are known as dictyosomes. Okay. So now let us see the structure of this Golgi apparatus. So as I told you that the Golgi apparatus is made up of the tubules, vesicles and cisternae. So these long tube-like structures are known as the tubules. The end portion of these tubules forms a bulb-like structure. These are known as the cisternae. And then these bulb-like structures get detached. They are the newly formed vesicles and they are known as vesicles. Okay. So from these tubules, these cisternae start developing, these bulb-like structures start developing, and slowly as they develop, they get detached from the tubules and they form the newly formed vesicles. Okay, so this Golgi apparatus appears in the form of three parts, that is, one was tubule, another was the cisternae, and the third was the vesicles. About the cis phase and the trans phase of the Golgi apparatus, you will be studying it in detail in your higher classes. In class 11th, you will be studying it in detail, but for now you have to study this only, that the Golgi apparatus is made up of three parts, that is the tubule portion, the cisternae portion and the vesicles. Now what is the function of the Golgi bodies? They help in the synthesis of certain polysaccharides from simple sugar. They also form the glycoproteins. They also help in the formation of glycoproteins. And then it is also involved in the transportation or transformation of these polysaccharides and glycoproteins. Now, the next function of Golgi bodies is that the primary lysosomes are also formed from the Golgi bodies with the help of budding. We will be studying about the lysosomes in the next slide, and they are also responsible for the secretion and transportation in the cell. So what they are doing, they are helping in the formation of polysaccharides, they are helping in the formation of the glycoprotein, then they are also forming the lysosomes, and then they are also actively participating in the secretion and transportation of the materials inside the cell. Now comes the next organelle, that is the lysosomes. So what are lysosomes? These lysosomes are small sized structures which are surrounded by a single membrane structure, and these lysosomes contain certain hydrolytic enzymes. Okay. Now, during the disturbance in cellular metabolism, like if anything mishappened inside our body, that is some wear and tear is going on, or if certain cells gets damaged, then what happens? These lysosomes can burst, and then after bursting, they digest the content of that particular cell. Okay, so inside the cell many organelles are present, and one of those organelles are the lysosomes. Lysosomes are containing certain hydrolytic enzymes, and if in case inside that particular cell in which this lysosome is present, there some mishappening happens and the cell gets damaged, then these lysosomes burst up, they get opened, the enzymes present in them comes out, it digests their own cell then. Okay, then they help in the digestion of their own cell. Therefore, due to this function of the lysosomes are also known as suicidal bags of the cell. They ultimately lead to the suicide of the cell in which they were present. Okay, so enzymes which are present inside the lysosomes, they are made by the rough endoplasmic reticulum, and they also help, the lysosomes also help in the destruction of the foreign materials if it enters into the cell. So now this is the structure of the lysosome. It is the single walled structure, single membrane is there, and then there are many different enzymes which are present, hydrolytic enzymes which are present inside the lysosomes. Okay, so in the cell you can observe lysosome in this way. These are small circular structures which are present into the cell, which contains certain hydrolytic enzymes, and they act as the suicidal bags of the cell. So now the function of the lysosomes. So they help in the digestion of the food within the cell, that is intracellular digestion. Inside the cell, whatever digestion has to take place, it takes place with the help of these lysosomes. Then again, they help our body from the attack of the bacteria and harmful organisms. Whatever bacteria or harmful organisms or foreign particle enters into our body, lysosomes digest them, and they also helps in the removal of the old worn out cell organelles. If in case inside the cell certain organelles are not working, they have gone old, or the cell itself gets damaged, then these lysosomes helps in cleaning up of that particular content of the cell which is not working properly. Okay students, so we will stop here for today. Thank you very much.